Our next presentation is presented by Rahul Goyal. He's a PhD candidate at the University of Maryland School of Pharmacy for Pharmaceutical Sciences, and his presentation is ScreenMCM, a machine learning-based product screening tool to accelerate medical countermeasure development. Thank you so much.
Hello everyone. Thank you for taking the time to listen to my talk. Today I will be talking about ScreenMCM, a machine learning based product screening tool to accelerate medical countermeasure development. Precision medicine is usually implemented using clinical decision support systems. In saying precision medicine, I refer to precision pharmacotherapy here. The following three components are at the core of these decision support systems: data, data analysis, and application software. Achieving precision medicine is a continuum, meaning it cannot start after approval of the drug. Precision medicine should start through drug development and it is used through the clinical practice. Thus, the data for these tools can come from experiments that are performed before approval of the drug or from real-world data post-approval. This data is analyzed using different data analysis methods, and then a software that can execute this data analysis methods is created, leading to a clinical decision support system. Currently, in terms of data analysis, statistical and mechanistic approaches are used. However, with increasing volumes and varieties of data, innovation of new methods is needed. In this context, machine learning acts as a new and advanced way of analyzing data that can be seamlessly integrated into application software to achieve the goal of precision medicine. Usage of machine learning is same, be it in precision medicine or drug development. I will show an application of machine learning in drug development, but the same can be used in precision medicine. Allow me to show how machine learning can be used to create a useful solution in drug development by starting with some background for my case study. Exposure to high doses of ionizing radiation in a short period of time can occur due to treatment, occupational hazard, or terrorist incidents. This exposure leads to a disease called as acute radiation syndrome, or ARS. In ARS, the bone marrow is affected. That leads to lower than normal number of mature blood cells, which leads to mortality. To counteract ARS, treatments called as medical countermeasures or MCMs are developed. On a high level, if a company has a lead product in development that they think can be a potential MCM, it is tested in animals for its effectiveness in treating ARS, usually by showing a significant decrease in mortality rate. These products cannot be tested in humans due to feasibility and ethical reasons. Once a product is shown to be effective in animals, it is then approved by the FDA as an MCM. However, do you know what the failure rate of products is when they are tested in animals? It is 75%. This means that we do not have a good screening tool that can tell us the effectiveness of a product before we actually test it. Having better screening tools can reduce this failure rate. Let me show how we developed a product screening tool with biologic understanding of the disease and machine learning that can potentially cut this failure rate in half. Our tool, ScreenMCM, can screen new products in less than 1/3 of the time that it takes currently. ScreenMCM intends to reduce trial duration of proof of concept trials and their respective sample sizes. The utility of such a tool also falls in line with the recently passed law which authorizes use of computer models for investigating effectiveness of a drug. Now that I have shown what the tool can potentially do, I will go over how it was developed and also an application in screening products. The tool was developed by using existing data from studies that were already performed. These studies are usually looked at in silos. However, through this research, I am able to pool information and extract knowledge that can accelerate the development of medical countermeasures. I have big data for more than 500 animals for all hematologic biomarkers collected at different radiation doses and days, and using this data, I aim to develop a product screening tool that can take as inputs different effects a new product has on different hematologic biomarkers and predict if the product is effective or ineffective in reducing mortality rate in patients experiencing ARS. I would first like to briefly touch upon the details for the studies. The data contained three 60-day studies. Studies 1 and 3 were used for the FDA approval of the MCM leukine, and study 2 was an exploratory study to test a delayed treatment approach for leukine. The study arms are listed here. For example, V1 24 hours means that in this arm, the vehicle article was administered 24 hours post-irradiation. L1 24 hours means that in this arm, leukine was administered 24 hours post-irradiation. After pooling data from all studies, there was data for biomarkers in the red blood cell lineage, platelets, and white blood cell lineage, collected every day for first 30 days for 500 plus animals, and due to the longitudinal nature of the data, this resulted in approximately 15,000 rows. Now I will show my approach towards modeling mortality due to ARS. This approach consisted of 5 steps. First, I started with data preparation, where the inputs to my model are raw levels of biomarkers on different days or aggregated level of biomarkers over a time window. And the outcome was a binary variable, that is 60-day mortality. Once data was prepared, 60% of it was split to train the machine learning models and 40% was split to test the prediction performance of the final model. Once the data was split, using the 60% training data, different machine learning models such as elastic net regression, XGBoost, support vector machines, and random forest were fitted. Once the models were fitted, the models were qualified using several performance metrics such as area under the ROC curve, accuracy, calibration plot, and Spiegel-Halter z-test. Upon qualification, the final model was tested for its prediction performance on unseen data, that is 40% of the testing data that was set aside. Now I will show my approach towards data preparation. On the left, I have the trend for platelets, which is the case for most biomarkers, and on the right, I have percentage of decedents by study day. Some points to observe here are that biomarker depletion started around day 4, recovery started around day 13, and that 95% of the animals that died between days 12 and 24. Therefore, I chose a time frame of days 4 to 10 because depletion for most biomarkers occurs during that time frame, and I can also develop a model that can predict mortality into the future. Now that the time frame was selected, to aggregate this data for modeling, I used different metrics listed here such as area under curve, slope, maximum, minimum, and so on. The aggregation resulted in the final data set consisting of 500 animals with 136 predictors. After splitting this dataset into training and testing and fitting different models, I moved to model qualification. Model qualification results are shown in this graph. On the x-axis, I have 4 models: elastic net regression, random forest, support vector machines, and XGBoost. On the y-axis, I have prediction performance and 95% confidence interval for accuracy and area under the ROC curve. One can see that all models demonstrated equivalent performance. Therefore, I chose the most simplistic model to go ahead with, that is the elastic net model. Using this model, I tested it for model calibration by comparing the predicted probability of mortality to observed event rate, and you can see that both of them matched well. The Spiegel-Halter z-test also resulted in an insignificant p-value, suggesting that the model is well calibrated. Using the final elastic net model, I performed recursive feature elimination and obtained 19 features in the final model with neutrophils and platelets as the top predictors of mortality. Similarly, in a hospital setting for a patient undergoing vancomycin therapy, what if we had neonate status, kidney function, drug exposure, concomitant medications, and other patient characteristics, and we wanted to know the risk factors for kidney injury to optimize vancomycin dosing? We can put this information into a machine learning algorithm using the same methods that I showed here and predict when the patient might experience kidney injury to individualize dose. I will be moving forward now and show the result for my next and final step in model development, that is, testing the prediction performance on unseen data. The prediction performance on new data was found to be adequate, with both accuracy and area under the curve being greater than 0.7. This model and its prediction performance thus gives us confidence to apply this tool for product screening. Now that the tool is adequate for prediction purpose, let's see how we can apply this tool for screening products with a prototype and a simplistic approach where the products affect only one biomarker. Let's say the sponsor has data from 4 products from a total body radiation model or an in vitro study to understand effect on hematopoiesis. And we want to see which of these products we want to move forward with for development. These products have different effect sizes on daily platelet value. For a given radiation dose, let's say that the expected survival rate for placebo is 23%. Now I will apply this effect size of the different products into ScreenMCM. ScreenMCM shows expected survival rate for all of these products. Based on the predicted survival rate, the sponsor would decide to move ahead with product 4 for development. The application here is a very simplistic case. ScreenMCM can help with answering any sort of combination. For example, product 1 might improve platelets and neutrophils. Product 2 might improve neutrophils and red blood cells. Product 3 might improve only platelets. We can translate all of this information into expected survival rate for each product. This property of the tool can potentially help tailor products in development. Finally, the predictions shown here will also have uncertainty for the survival rate, but I'm only displaying the mean for the sake of simplicity. The uncertainty can help design registration trials for the product. To conclude, I have shown how a machine learning tool, namely ScreenMCM, was developed that can accelerate medical countermeasure development by achieving the following 4 results. ScreenMCM can increase probability of trial success because it can accurately predict the net treatment effect of a given product before actually testing it. ScreenMCM can shorten product development time because with data collected from only a 10-day study instead of 60 days, one can gain information on product effectiveness. ScreenMCM can also help with reduced animal testing because with such a tool, studies are not required to capture all the variability in mortality, but rather shorter studies with fewer animals that can accurately capture biomarker levels are sufficient. Finally, because ScreenMCM is a machine learning tool, it can continually improve with more knowledge that is available with regards to other variables such as inflammatory biomarkers or immune system modulating biomarkers. Lastly, I wanted to highlight some examples from literature to show how machine learning is being used in precision medicine. Approach in these examples is same as I demonstrated in my case study. We identify the relationship between biomarkers and outcomes, and we can use it in real time as new data becomes available. In this first example, a machine learning model that can take as input inflammatory cytokines, patient characteristics, and treatment plan was developed that could predict an adverse event. This model can be used to individualize radiation therapy in patients with non-small cell lung cancer. In another example, a machine learning model was used with radiomics to develop a CT image biomarker that could predict treatment response in patients receiving anti-PD-1 or PD-L1 immunotherapy. This biomarker can not only be used to individualize treatment, but also can be correlated with other clinical outcomes in this patient population. I would like to acknowledge Partner Therapeutics, who sponsored this project, and BARDA, who sponsored the 3 studies whose data was used for this analysis, and also my collaborators, Dr. Gopalakrishnan and Dr. Goburu from the University of Maryland, and John McManus from Partner Therapeutics. Thank you.